Hello everyone, welcome to our demonstration video on SDSPH or sodium dodecyl sulfate polyacrylamide gel electrophoresis. SDSPH is crucial because it allows to separate and analyze protein based on their molecular size. It is widely used for applications like protein purification, molecular weight determination, and even disease diagnosis. But how does SDSPH work? Let's break it down. Proteins are naturally complex with unit shapes and charges. To separate them based on size, we first treat them with SDS, a detergent that coats protein with a uniform negative charge and unfold them into linear shape. This way, the separation is purely based on molecular weight, not shape or charge. When an electric current is applied, the negatively charged protein migrates through the polyacrylamide gel matrix towards the positive electrode. Smaller proteins move faster because they can easily pass through the gel pores, while larger protein moves slower. Common methods used in SDS are gel casting, sample loading, electrophoresis, and staining. While SDSPH is a fundamental technique, it's often combined with other methods for a more comprehensive analysis. This method includes Western blotting to detect specific protein using antibodies, mass spectrometry for precise protein identification and structural analysis, and enzyme activity assay to study the function of protein. The instruments needed to perform SDSPH include the gel casting apparatus, electrophoresis chamber, power supply, micropipettes and tip, and staining equipment. SDSPH can analyze various types of protein samples. These include purified protein, cell lysate, tissue extract, and enzyme preparation.
SDSPH is used to protein separation based on the size. The gel is made of polyacrylamide and consists of two layers, which is the resolving gel for separation and the stacking gel for sample alignment. The material needed is acrylamide to form the gel, SDS to denature the protein, tris buffer for pH control, APS and AMP, TEMET, and the last one is gel casting plates, comb and pipettes. Next is step for gel preparation. The first step is to making the resolving gel. By mix acrylamide, tris buffer in the pH 8.8, SDS, water, APS and TEMET. Then pour into the gel plates with 3/4 full and overlay with isopropanol of water to prevent drying. Let it polymerize for 20 to 30 minutes. Next is making the stacking gel. By mix acrylamide, tris buffer with pH 6.8, SDS, water, APS and TEMET, and remove isopropanol from the resolving gel. Pour stacking and insert the comb. Let it sit 15 to 20 minutes. Next is the type of gel. The first one is resolving gel, 8 to 15% acrylamide with the pH 8.8 for separation the protein. And second is stacking gel, 4 to 5% acrylamide in the pH 6.8 for protein alignment before separation. For safety precaution, we need to wear glove and lab coat because the acrylamide is toxic. Work in ventilated area to avoid fumes. Handle APS and AMP TEMET with care to avoid irritation. And the last one is to dispose the waste properly by do not pour it into the sink.
Assalamualaikum, my name is Nurin Farisha binti Khairus. I would like to explain about how to prepare the protein sample in SDSPH. First, take the sample such as bacterial cell pellet and protein. After boiling, cool the sample on ice to prevent any degradation. Before loading the sample, it is important to know the protein concentration. Then, once the protein is prepared, load the appropriate volume into the wells of the SDSPH gel. After loading, the gel is run in an electric field and protein migrate according to their size, with smaller protein moving faster through the gel matrix than larger one. By the end of the experiment, we will use a molecular weight marker, also known as a ladder or standard, as a component to determine the size, of protein or nucleic acid fragment to run on a electrophoresis gel. Tips on how to avoid the sample contamination is by using a proper handling tools such as a wear glove and use tweezer or other handling tools to reduce risk of contamination from hand to tools and sample. Sample denaturation, the process of disrupting the folded structure of a protein or nucleic acid molecule within a sample. This is crucial for ensuring that proteins are separated based purely on their size and not based on their natural shape or charge. Denaturation helps break down complex structure and give protein a uniform negative charge, allowing them to migrate properly during electrophoresis.
Hello, my name is Muhammad Zakwan and I'm gonna explain to you about electrophoresis process. Electrophoresis is a laboratory technique used to separate charged molecules such as proteins or nucleic acids based on their size and charge. In this process, a gel matrix, usually polyacrylamide or agarose, is prepared and submerged in a buffer solution within an electrophoresis chamber. The samples are loaded into wells at one end of the gel and an electric current is applied across the gel, typically with the negative electrode, which is cathode, at the sample loading site and the positive electrode, which is anode, at the opposite end. As the current flows, charged molecules migrate through the gel toward the electrode with the opposite charge, allowing for separation based on size and other properties. Smaller proteins encounter less resistance in the gel matrix and travel faster toward the anode, while larger proteins move more slowly due to the greater friction with the gel fibers. This results in size-based separation, where proteins form distinct bands that can be visualized through staining techniques like Kumasi, brilliant blue, or silver staining. So, what are the factors influencing protein migration? Several factors affect protein migration during electrophoresis, including voltage, gel concentration, buffer composition, and sample preparation. Higher voltage increases migration speed but may lead to overheating, which can distort bands or degrade proteins. The gel concentration determines pore size, which is higher acrylamide concentration creates smaller pores and better dissolving smaller proteins, while lower concentrations allow larger proteins to migrate more efficiently. The choice of buffer maintains pH and conductivity, ensuring stable migration. Additionally, sample preparation such as denaturation with SDS and reduction of disulfide bonds with DDT ensures consistent protein preparation. And lastly, this is an important part, which is monitoring and ensuring proper running condition. To achieve optimal results, the electrophoresis process must be carefully monitored. The progress is typically tracked using a visible tracking die, such as bromophenol blue, which migrates at a predictable rate and help estimate the runtime. Proper conditions include maintaining a stable voltage, preventing overheating by using a cooling system if needed, and ensuring the gel is properly polymerized and submerged in a correct buffer. And after the run, proteins are stained to visualize their position, and additional techniques like Western blotting may be used for further analysis.
Hello everyone, my name is Farah Atila and I am going to conclude everything that my friend already said about SDS pitch. So, in summary, SDS pitch is a powerful technique widely used in molecular biology and biochemistry for protein separation based on their size. It plays a crucial role in protein analysis, helping researchers to identify and study protein in various biological samples. The process begins with gel casting, where polyacrylamide gels are carefully prepared to create a medium for protein separation. Next is sample loading to ensure that protein sample, along with molecular weight markers, are correctly placed into the wells for accurate migration. And lastly, electrophoresis that applies an electric field by allowing the protein samples to move through the gel and smaller proteins traveling faster compared to the larger one. Ultimately, STS-PAGE remain as essential tools in biochemical studies, aiding in disease research, drug developments, and also various scientific discoveries. Understanding each step of the process ensures accurate result and reliable data for further application in molecular biology.